Hello, everyone. Thank you for joining us. I'm Steve,
And I'm Scott.
and together we are Backyard Musings. We are broadcasting live from the backyard.
Yep, a little
Yes,
chilly this morning.
yesterday it was the front yard because we had missionaries playing volleyball for their P day.
Yeah, they were out of control.
They were they were having fun.
Yeah,
They get what 8 hours to have fun
I
per
think so.
week.
Yeah.
Yeah, crazy.
Well, they're calling us fun, but
Yeah,
they to let their
hair
down, so to
Yeah.
speak. So
Yeah. So we got an update on cancer. So we saw a Fox News video about this Navy veteran. He's retired Navy. Name is John Ryan, and he had stage 4 carcinoma in the lungs, diagnosed in 2013. And Walter
not
Reed,
good.
yeah, not good. Yeah, Walter Reed Medical Center. That's the big. It's
Military hospital, right?
yeah, military. I think it's naval in Bethesda, Maryland. Yeah, but it's all military. They couldn't help him. He got a second opinion. Hey, check out Johns Hopkins. So he checked out Johns Hopkins, Dr. Julie Bramer, and he went into a clinical trial, 2-year trial for immunotherapy. And then somewhere in there, he said 100 months. That's 8 years. So I don't know. Maybe I don't know what the 100 months was, but he said 2 years. The trial was 2 years. And so what they would do is during a treatment, they would freeze part of his lung, and then they had to give him something so that the
some
blood
kind of
clots,
injection, right?
yeah,
Some kind of injection
to prevent the blood clots, and then another, they call it CBRT, for something to drain something. And he said after 4 treatments, he had 64% shrink of the carcinoma. Huge. And then it eventually went to 82%, then 84%. I mean, just huge.
Miraculous.
Lung cancer. Right? Miraculous.
Yeah.
And he talked about that faith. He talked about having faith. But we're on the cutting edge of this cancer technology. I don't know if you'd be able to do that with a lymph or a throat, esophageal,
or a gland, like
or a gland,
or something like that.
But for the lungs, that seemed to work. That immunotherapy is what he called it.
It worked for this guy. So whatever. That's
exciting.
We like that kind of story.
Exciting.
Definitely. Yeah. And we've been talking about a lot of cutting edge stuff involving cancer, right?
And Alzheimer's and dementia.
Right? Yeah.
There's been a couple articles. I think we've been inundated on our show with Alzheimer's, so I backed off a little bit. But there's been some different breakthroughs on Alzheimer's. Maybe we'll come around circle and talk about them. Yeah, but yeah, they're pushing the envelope.
Yeah, we like it.
Say. All right, a research team from Osaka University investigates the impact of antioxidant enzymes on specific cell types and their role in age-related diseases. Age-related would be dementia, Alzheimer's. A recent study published in Blood by researchers from Osaka University and other institutions in Japan explores the critical antioxidant role of selenoproteins. The study examines how disrupting selenoprotein production impacts different cell types and hematopoiesis. Did I say that right?
Sure.
Hematopoiesis?
That's one of the words I was talking to you about, so go ahead.
The process of blood cell formation. Would you like to know more?
Many foods are marketed for their antioxidant benefits, which help neutralize reactive oxygen species, ROS, highly reactive molecules that can damage lipids, proteins, and DNA in human cells. Excess ROS accumulation is linked to age-related diseases, including cancer, highlighting the need to maintain a balanced antioxidant system. Human cells have 25 different selenoproteins. These antioxidant enzymes also convert dangerous ROS, such as lipid peroxides, into a safer form. That's good, right?
Yeah.
Buildup of lipid oxide or peroxides can affect critical cells called hematopoietic stem cells. Okay, a phenomenon observed in aging diseases.
Now, I didn't know anything. 23 selenoproteins. I don't know what a selenoprotein is.
I did not know either. Antioxidants, I've heard of that kind of stuff, but and again, it mentions in that it talks about age-related. There's a lot of cancers that are not age-related, right? I mean, all you got to do is watch one of those commercials.
Colon cancer.
Yeah, but that one,
esophageal.
St. Jude's. Oh my gosh, those are tearjerkers. These kids.
Oh yeah, the
that are
kids. Yeah.
going through that stuff. So, but
Yeah.
anyway,
That is tearjerker.
it is.
All right, quote, we observed that aged HSCs frequently display impaired selenoprotein synthesis, but it was unclear how this could contribute to cell aging and if it could be reversed, end quote, says Yumi Aoyama, co-lead author of the study. Quote, we hypothesized that selenoproteins are a critical part of the antioxidant system that fights age-related changes in HSCs. To investigate this, the team used a mouse model. Mice with a certain gene knocked out, leading to, I wonder what that certain gene is, leading to disrupted selenoprotein production. They then examined how this affected different cell types, finding that the knockout negatively impacted HSCs and immune cells with B cell lineages, types of white blood cells, but had few effects on myeloid cells, a different family of immune cells.
Okay, this is a quote. The most notable results of the knockout included B lymphocytopenia, which means there were fewer B cells than expected. Explained by Hiromi Yamazaki, the other co-lead author. Again, quote, the HSCs are also had a limited ability to self-renew. These observations, along with increased expression levels of aging-related genes in those cells, were consistent with what is frequently seen in age-related diseases. Further investigation indicated that the effects were driven by lipid peroxidation. Additionally, experiments with cells from the mouse model revealed that the disruption in selenoprotein synthesis could support B progenitors switching to the myeloid cell family. So
that's like,
this is.
So it's good cutting edge.
Obviously, this is not something you're going to treat with Bactine.
Right.
Yeah, this is a little bit.
Right. Here's another quote. Our data suggests clear lineage-specific effects when the protective role of selenoproteins is lost, says Daiichi Inoue, senior author of the study. Quote, these enzymes are critical for counteracting the lipid peroxides that accumulate during the aging process. The researchers also investigated the mechanisms underlying hematopoiesis with a feeding experiment on the knockout mice. They revealed that dietary vitamin E can protect hematopoiesis and has the ability to repair impaired B cell differentiation.
Okay.
All right.
We'll take their word for it.
Yeah.
This study shows the antioxidant functions of selenoproteins and how they ensure proper HSC self-renewal and B cell lineage immune cell maturation. Because the knockout mice displayed similar phenotypes to aged normal mice, the findings demonstrate how potentially addressing selenoprotein production related issues could help fight age-related diseases. So
I wonder what is the age range of a mouse? I mean, do they live for a month or a year? And I guess this would help with age-related diseases,
Well, I don't know. In a lab setting, maybe they can prolong their life. If there's a rodent like that found in my house, it's ours. Yeah. If it's found, it's hunted and exterminated.
I'm like, in the lab.
I'm sure they can probably prolong these mice because they've got to do these tests, right? So they want these mice to live as long as possible.
Yeah.
And better a mouse than some other type of animal. We don't want any of that kind of stuff. But I don't know. That would be an interesting.
This is interesting
thing to investigate, I guess. Right.
Hopefully, this leads to something that can help impact Alzheimer's or dementia. Or even cancer.
A good majority of the stories that we talk about have mice models.
Yeah, definitely.
So
yeah,
it's good.
All right, folks. Thanks for joining us.
Take care, everyone. Have a good day.